Biology 9700, and the topic is protein synthesis. And students, you know the DNA is a code for protein, and however DNA is found in the nucleus and proteins are made at ribosomes. And you know the DNA is present inside the nucleus of a cell which control all the activities, and ribosomes are present freely available in the cytoplasm, or they are present on the rough endoplasmic reticulum. So the information has to be reached from the DNA to the ribosomes. So there must therefore be some way of getting information from the DNA to the ribosomes. The solution to this problem is to use an intermediate molecule that carries the information is messenger RNA. So the messenger RNA brings the information from the DNA to the ribosome where the protein is synthesized. So DNA makes RNA and RNA makes protein. So protein is synthesized in two steps. The first process by which DNA makes messenger RNA is called as transcription. So the first step in the protein synthesis is transcription, in which DNA is copied and messenger RNA is formed. The process by which the message carried by messenger RNA is decoded to the making of protein is called translation. And the second step, when the messenger RNA is decoded at the ribosome, and then the protein is formed. That is, that process is called as translation. So, students, you can see the first step, the transcription, it takes place in the nucleus. So, it takes place in the nucleus, whereas the other step, which is translation, in which protein is synthesized at ribosome. So, it takes place in the cytoplasm. So both of these steps are responsible for the synthesis of protein. Now, the first step, which is called as transcription, and you know the transcription takes place in the nucleus. So transcription takes place in the nucleus. The enzyme responsible for transcription is called RNA polymerase. And students, you know, all the chemical reactions or all the metabolic reactions which are taking place inside the cell, all they need the enzymes. So here in this case, the transcription has to be taken place. So the enzyme which is responsible for this purpose is called as the RNA polymerase. So the RNA polymerase attaches to the beginning of the gene to be copied. So the RNA is responsible for the synthesis, for the process, or the first step, which is transcription. It starts to unwind the DNA of the gene, and another enzyme breaks the hydrogen bonds between the two strands or unzips the DNA. Students, you know, the DNA is a double helical structure, and first of all they unwind, and they just like a ladder. When they are unwinded, they are just like a ladder. So what happened? First of all, they are unwinded, and when they are unwinded, so you know the hydrogen bonds are present between the nitrogen bases. So these um, hydrogen bonds start breaking, and these two strands get separated from each other. So these two strands get separated from each other. So this creates two single strand sections of DNA with the normal double helical structure either side of the unzipped section. And you can see in this way, only one of the exposed strand is copied. So any one of this strand will be copied by the messenger RNA. So this is called the template strand or transcribed strand. Any one of them strand will be copied. So our complementary RNA copy of the template strand is made. Remember that RNA contains the base uracil instead of thymine. This means that the bases adenine, guanine, thymine, and cytosine in the DNA are copied in the RNA as uracil, cytosine, adenine, and guanine. And students, you know that these are the four nitrogen bases are there, which are present in the DNA. They are adenine, which combines with the thymine, and guanine with the cytosine. Adenine and guanine are the purines with double rings, and thymine and cytosine are the pyrimidines with single ring. So one purine combines or binds with the one pyrimidine. So A always combines with thymine with double bond, and double bond, these are the hydrogen bonds. And guanine, when it binds with the cytosine, there are three hydrogen bonds between them. So two hydrogen bonds when adenine and thymine, and when guanine combines with the cytosine, there are three hydrogen bonds. Now, you know that students, there is a difference between DNA and the RNA. In DNA, thymine is there, but in case of RNA, instead of thymine, uracil is there. So adenine in RNA, adenine will join with the uracil instead of thymine, because thymine is only present in DNA and in, but all other three nitrogen bases will be the same. Only there is replacement of thymine, the uracil is there. So during this first step or transcription, uh, the code is a three-letter code which is called as triplet code. Each set of three basis codes for one amino acid. And students, you know, three nitrogen bases. Three nitrogen bases. They will make up a one code, single code, which is called as triplet code. Triplet code because it is made up of three nitrogen bases. As for example, triple A, adenine, 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 or uh, or it can be cytosine, adenine, thymine, or it can be thymine, adenine, cytosine. So there are many examples of these different types of, you can say, amino acids. So three nitrogen bases will make up one amino acid. Three nitrogen bases, any one of three, they will make up one amino acid. And you know, the 20 naturally 20 different amino acids are available in the nature. So the lower stand, each set of three bases code for one amino acid. The lower stand is the template stand. Any one of these strands which will be copied by the messenger RNA will be called as the template stand. Reading from the left hand end of the template stand, for example, the first triplet shown is cytosine, adenine, adenine. So this is an example which can be given, but that is then if we read on the first three triplet codes uh, in the template stand, it can be CAA, and CAA stands for an amino acid which is called as valine. So messenger RNA is made from nucleotides found free in solution in the nucleus. As the RNA polymerase moves along the gene, the nucleotides approach and hydrogen bond with their complementary nucleotides in the DNA. So as you can say, the DNA strand, it is the portion which has to be copied. The, the complementary bases, they copy this part of the DNA, and then as each nucleotide arrives, RNA polymerase joins it to the growing messenger RNA with the polyphosphodiester bonds. And you know, the phosphodiester bonds are present between them. Once phosphodiester bonds are formed, hydrogen bonding of that part of the messenger RNA to the DNA is no longer necessary and the hydrogen bonds are broken. And you know the hydrogen bonds between which are present between the nitrogen bases of the DNA, they will be broken down, because when they will be broken down, then the DNA strands will get separated from each other. Eventually, a stop message will be reached. Stop message which will stop the process of transcription. So, because it will show the part of the DNA which has to be copied, which has to be transcribed. It is completed. No, the stop message will be there. At this point, the RNA polymerase releases the completed RNA. It means the messenger RNA has been completed and the RNA polymerase leaves the DNA. Now, the first step has to be completed uh, in the nucleus, and then when the RNA polymerase has completed the formation of messenger RNA, then after this it has to leave the DNA, and after this we can see there is some type of modification which is needed to be done in the messenger RNA. After this, the messenger RNA will be completed and then it will leave the nucleus. So in eukaryotes, the messenger RNA is modified, partly changed before it leaves the nucleus, the original molecule before it is modified. Modification is necessary. And when without modified, it is called the primary transcript. So the process of modification is called RNA processing. Process of modification, very simple. It is called as DNA RNA processing. So one step in this processing is RNA splicing. So during RNA splicing, splicing is the removal of section of primary transcripts. The sections removed are called introns, non-bonding parts. So as are the sections of DNA that codes for them, they are not the parts of the RNA. So they are only the part of DNA. So they are removed. So the nucleotide sequence that remains after the introns are removed are called exons, and the remaining parts of the are called as the exons. They have to join together after removal of the introns. So they will join together with the help of bonds, and then after this modification, the messenger RNA will leave the nucleus. It will move out the nucleus to the nucleopore, and then it will enter, into the cytoplasm. Now here you can see the whole process which I have described in the form of a diagram. Here you can see DNA strand, double helical or DNA strand, and you can say the process is from this direction towards that direction. So here you can see this is the polymerase, RNA polymerase on which the DNA strand is attached, and then unwinding of the DNA strand has been started, and in the place which has been copied, the part of the DNA which has been copied. So at that place, the rewinding of DNA has been started. So rewinding has been started, and the other part remaining, you can see the red color. With red color has been shown the RNA which has been copied from this template. So the lower strand will act as a template strand, and from this template strand, this messenger RNA has been formed, and this is the region where this is transcription is taking place. That portion is called as RNA DNA hybrid region, and then there is unwinding of DNA. Now unwinding of DNA has been taking place over here. So nucleotides being added to the 3 and of the RNA. So in this way, the two strands, first of all, they will unwind, then messenger RNA will copy the template strand. And here in this case, the lower strand is acting as a template strand. And after copying the messenger RNA, when the transcription is completed and modification has been taken place into it, then this messenger RNA will move out of the nucleus to nuclear pore and it will enter into the cytoplasm. So this is the first step, which is called as transcription, and it has been shown in the form of this diagram, and you can clearly see the unwinding of the DNA molecule and the copying of the messenger RNA or the formation of the messenger RNA. And then after the formation of messenger RNA, there is rewinding of the DNA molecule, and then messenger RNA is ready to move out of the nucleus and it enters into the cytoplasm, and where it will attach itself on the ribosome. And the second part of the protein synthesis will take place, which is called as translation. And the second step is translation, which takes place in the on the ribosome, which are present not only in the cytoplasm, but they are also present on the endoplasmic reticulum. So after the transcription of DNA to messenger RNA is complete, translation or the reading of these messenger RNAs to make proteins begins. So the protein synthesis will start, or the making of the protein will start right now after the uh, transcription, or when messenger RNA is reached into the cytoplasm on the ribosome. Recall that messenger RNA molecules are single-stranded. And yes, the DNA is double-strand molecule, whereas the DNA is double-strand molecule, and whereas RNA is the single-strand. So adenine, uracil, cytosine, and guanine is complementary bases, that is specific portions of DNA cell's DNA. Each messenger RNA dictates the order in which amino acids should be added to a growing protein as it is synthesized. In fact, every amino acid is represented by a 3-nucleotide sequence or codon. Already told you, the triplet or triplet codon, it will represent one amino acid which consists of 3 nitrogen bases. For example, AGC. AGC is the messenger RNA codon for the amino acid serine, and UAA is a signal to stop translating a protein is called the stop codon. Again, when there will be the, you can say the translation is completed after the formation of a protein, that is called as UAA is the signal or the stop codon, after which the translation will be stopped. Now, it means the after translation. Now, here you can see in this case, when there is protein is manufactured during this protein synthesis, the molecules of transfer RNA, here the messenger RNA, it means all 3 types of RNAs are involved in protein synthesis. Messenger RNA, which is copied, and then it moves from the nucleus to the cytoplasm, where on the ribosome it is attached, and in that case the transfer RNA are responsible for matching amino acids with the appropriate codons in messenger RNA. So transfer RNA will brings the amino acids or triplet codes in the form of triplet codes on the ribosomes. So each complementary basis, each transfer RNA molecule has 2 distinct ends, one of which binds to a specific amino acid. As for example, you can say they will be attached in this way, on which 3 nitrogen bases, as for example, CAT or CAA, we can say CAAAA. It codes for valine. So this is the structure of the ribosomal RNA, and on one side The three nitrogen bases are attached on the other side. You can say the amino acid is there. So each transfer RNA molecule has two distinct ends, one of which ends to a specific amino acid, as I have shown valine, and the other which binds to the corresponding messenger RNA codon, CAAA. So during translation, these transfer RNAs carries amino acids to the ribosomes and join with their complementary codons. So the translation will start from one end and it will be completed. Then the assembled amino acids are joined together as the ribosomes with its resident ribosomal RNA, the third RNA, which is present in the ribosomes. It will help in the arrangement of the amino acids. So moves along the messenger RNA molecule in a ratchet-like motion. The resulting protein chains can be hundreds of amino acids in length, and synthesizing these molecules require a huge amount of chemical energy. So as for example, this is the messenger RNA, and on messenger RNA, the nitrogen bases are present in the three triplet chords. So in this way, they, as I have shown, in this way, they will be nitrogen bases will be tagged, and this is the amino acid, and in this way, they will start translation, and translation will start from one end, and after it will be completed. Now here in the next diagram, I would like to explain all which happens in the translation. And here you can see this is the messenger RNA, and messenger RNA has three different codes. You can see GAU is a one code, UCA another code, ACG another code, CGU. It means, so they have shown three different amino acid, ribosomal RNA, transfer RNA, number two transfer RNA, number three transfer RNA. The amino acids are there. So these amino acids which are joined on one end of this transfer RNA, and the other end you can see the triplet codes which are the anti. These codes will act as the codons, and the three nitrogen bases which are attached with the transfer RNA, they will called as anticodons. So codons and anticodons. You can say the uracil is present in the RNA. So uracil always joins with the adenine, guanine with cytosine, or cytosine with guanine. So in this way, the amino acids are present on one end. When it happens, when these nitrogen bases are joined at their complementary codons, then there is formation of peptide bonds between the amino acids which are present on other end of this transfer RNA. So after doing this, the transfer RNA are separated from one another, and in this way, a long polypeptide chains with thousands of amino acids will be formed. So, and all it happens, the translation process of translation, it happens on the ribosome. And students know this is the complete process of protein synthesis. After the synthesis of protein, it is transported through the endoplasmic reticulum, and from endoplasmic reticulum, it enters into the Golgi apparatus where it is modified, and then it is secreted out of the cell or inside the cell. It can be used. So in this way, it is all about the protein synthesis. Please like, share, and subscribe my channel. And thanks for watching.